All right, let's do let's see here. See, everybody disappears. Check 1 2, check 1 2, check 1 2, check 1 2.
Yep, I can hear you.
Good? Okay, good. I'll see how long this lasts. I'm not going to do the acrobatics today where I stand up on the platform. I think it might pull the cords apart. So, all right, cool. The Father, the Son, the Holy Spirit, amen. God, grant me the serenity to accept the things I cannot change, courage to change the things I can, wisdom to know the difference. Father, Son, Holy Spirit, amen. So let's get down to some business today. Got it. All right, let's do the normal thing here. We got Jasmine, Yo, Callum. Okay. Aiden. Okay. Matthew, Emily. That's Emily. Abigail. Abby, there you are. Ashley. Anna. Slowly. Donovan. Just do it. It's coming back into vogue. John Hurley. We got Quattro, there you go, Quattro. Good. Cameron. Rebecca. John Cost. Yo, is that the point? Okay, got it. Kate. Sophia, nice. Maria. Okay, there you go. Hey, Maria. Sam. Olivia. Got it. Christina. Kristen. Okay. Sarah. Tristan. Okay, Tristan. Okay. Caleb. Kyle. Jordan, yo, what's happening? Antonia. Yep. Deshaun, got him. Jonah, yo, what's happening? Landon. And Anthony. Good. Event next week. Starts with a Q, ends in a Z, has a we in the middle. Quiz. So, on the syllabus, I mistakenly wrote down that the quizzes will take place in here. They're not going to take place in here. They are going to be on Schoology. So quizzes will be available. You have 20 minutes, 20 minutes to complete them. And it's not open notes, it's not open book, it's not open internet. All right? They are, because we are in a realm of stress. Are you guys even allowed to use computers on campus? Is that like allowed, like different computer labs? Are you allowed to use those? Ish? Are you like kind of like encouraged to just stay in your room? Okay. Well, all right. So because of that, they will be resumable, which means if your service gets interrupted, you can go back in. Something happens, take a screenshot of what happened and just send it to me. This happened, blah, blah, blah. One of the worst things that happened, when you're taking a quiz, one of the worst things that can happen is for you to almost get to the end and then have your computer go, you're done. Or the internet cut off, okay? So take a screenshot. You should be able to go back in and fix it, resume where you were. So because it's resumable, that doesn't mean that you stop, go off, look up an answer, come back in. Don't do that, right? That's called cheating. So, but if something happens, send me a message, but take a screenshot of what happened. I want to see some evidence, right? Because science, we're all based, it's an evidence-based decisions. So I want to see some evidence that this happened. Send it to me. All right, it's good. All right. And remember that when we're in class, right, that means good. Okay. So, quiz. It's going to open Sunday at noon. Put it away. Thank you. Unless it's like from an emergency, we don't use that in here. Thank you. Okay, thanks. Sunday at noon until 10 o'clock Monday night. So it's open from Sunday at noon till 10 o'clock Monday night. You've got about 36 hours to take this thing. So if you wait until 9:50 and your battery isn't charged, not my problem. You got 36 hours to plan for it. You guys got this. You got this. This isn't a big deal. Everything's going to be, the questions will be based on these questions from the study guide. So hopefully you've been reading, you've been answering these. If there are some that you don't quite understand, hopefully you've been either reaching out to me, which nobody has, so I haven't gotten any emails from anyone, or you've been reaching out to the tutors. Remember, they are available on Schoology. There's a folder. See if I can actually find it. Nah, I'm not going to do that. That's going to wreck these guys. So there's a folder that says tutors, right? And there are links there for their Zoom sessions. It also gives you the hours when they're available. Good. So everything that we're going to finish talking about primary and secondary structure of proteins today. And so that will be the end of the material for the quiz. So let's review our amino acids here. We said there are 20, right? We said there's 20. Is an amino acid a monomer or a polymer? Ashley. Is an amino acid a monomer or a polymer? It's a monomer, right? It's just one. Good. Awesome. If I'm going to pull two amino acids together, I'm going to create a bond between those two. What's the unique name of the bond that holds amino acids together? Yes. Peptide bond. Exactly. Good. What kind of reaction? What are the two big categories of reactions we talked about on Wednesday? Yep. Okay, so we got dehydration and hydrolysis. Those are the two big kinds of reactions. So I'm going to form a bond. I'm going to use what kind of reaction? Hydrolysis, that's where we put the water on the pasta and it breaks it up. So how does the pasta start sticking together? Dehydration, okay? So remember, the pasta sits in the colander for too long, it forms a big, ucky lump. I just want a small scoop. Okay. He just put 5 pounds of pasta on his plate. Why? Because it's all stuck together, okay? So that's dehydration. Dehydration requires the input of Energy. Release of. Because it's dehydration. Sweet. So we form this protein. Good. All right. So we're still talking about amino acids. So we've got these big classifications of amino acids. We stopped. We talked about the non-polar ones. And remember that for all of these, hopefully you notice that for all of these amino acids, there are regions of the amino acids that are the same for all amino acids, right? They all have an amino group. They all have a carboxyl group. Amino at the one end, carboxyl at the other. In the middle, we have this little alpha carbon, which always has at least one hydrogen bound to it. And then we have the R group. This is what makes each of these unique. It gives each of the amino acids a unique structure, which then means that each amino acid has a unique in the protein, which is a macromolecule. Sweet, see, you guys got this. Polymer. So we said that if we looked at that, we first started out talking about the green, the blue, and the, what color would you guys call this? Rose? Mauve. Looks a little dusty rose. It looks like dusty rose to me. Well, let's talk about that in a moment. So, green. These amino acids, for the most part, the R groups are all made of which two elements, Kyle? You got it. The green ones, the R groups. What two elements do we primarily see in all of these? They're up on the screen here. Can you see that? It's kind of small, isn't it? Who can help them out? Carbon and, no. Yeah, there we go. Good, good, good. Yeah, the carbons look like oxygens. We're looking at the R groups. So when you have bonds between carbons and hydrogens, what kind of bonds are those going to be? Covalent bonds, but what kind of covalent bonds? Polar, non-polar? Anthony? Non-polar. Okay, we talked about that on Wednesday. That's important. So you can you should be able to easily identify a non-polar amino acid. I'm probably not going to give you these. So these ones here. There's a tricky one, which is tryptophan. Tricky tryptophan. It's tricky. It's an old song, tricky. Anyways, anyone ever heard of that song, It's Tricky? Okay. Anyways, I only know it because I'm OLD. Okay, so tryptophan is tricky because it has an amino group in it. Don't be tricked by that. That's the only one that's tricky. It's still non-polar. Then we moved on to the polars, the blues. Main thing that's important here is we see lots of amino groups, carboxyl groups, hydroxyl groups, carboxyl groups. If none of those words make sense to you, you better start reviewing your functional groups. Yo. All right. And then finally, we didn't get to the dusty rose ones yet, but let's do it. These are the charged ones, and these are really obvious. Why are they obvious? I'm going to call on some folks in the front row because it's hard to see Okay, Jasmine, why is it obvious that, that they're the charged ones? Okay, so they have a charge. Okay, good. That's right. These are charged. All right. So those are three major categories, right? Non-polar, polar, and charged. And then we have non-aromatic. Let's start with aromatic. What do we see about the structure of the R group on all of these aromatic amino acids? Who wants to take a shot at this? Name Ashley. Yeah. They all have rings. These are the three rings. These are the three Elvin rings. I'm just kidding. Okay. So the aromatic ones, they all have rings. The R group has a ring. All right, this has a ring. Don't worry about that. This is more, I'm really not going to ask you guys too much about these ones. So, good. So they all have rings. Rings oftentimes, the ring can actually slide in and stack in a protein. They're kind of cool. And then we have the non-aromatics. Do you see any rings up here? You ain't got no rings. You ain't got no rings. So that's non-aromatic. All right. I am mainly going to ask you about the non-polar, polar, and charged. So you should be able to identify, is this a polar, non-polar, or charged amino acid? All right. And you should be able to tell me if it can interact with water or not. If the R group can interact with water. So non-polar, well, we're going to talk about this in a little bit. But what about the R groups and these non-polar amino acids? What's the information that would tell me whether it can or can't interact with water? What's the big giveaway? What do we need to be able to interact with water? John? Polarity, either polarity or charge. So I don't see any polarity in these R groups, right? So are they going to interact with water well? No, they are not going to play well with water at all. They're going to interact with each other. We're going to see, once we start talking about structure, how that's going to impact the structure of the protein. What about these polar uncharged? Can they interact with water? Well, John just told us that it need to have either polarity or be charged to interact with water. So R groups of polar uncharged can certainly interact with water. And R groups of the charged amino acids absolutely interact with water. All right. Okay. So let's move on. Stop there for any clarifications, questions about the kinds of amino acids. Yeah, John. The charged ones, that's okay. The charged ones have a charge. So, yeah, and one good way we can actually look at this would be if we look at, let's look at asparagine here. So here we've got a carbonyl and an amine. See that at the end? We got a carbonyl and an amine. No charge. Aspartic acid. kind of close to that. But instead of having a carbonyl and an amine, we have a carboxyl group. And in this case, it's not just a carboxyl group, it's a carboxyl group that has lost its hydrogen. So that's why it's called aspartic acid. It's actually already donated its hydrogen. So it has a charge, negative charge. Okay? And just one other difference would be, again, let's compare asparagine. Now let's compare that with lysine. So let's look at the amino group. Here, the amino group, or the amine group on asparagine, has the structure, has the formula NH2. The amine group on lysine is an NH3 plus. It has an extra hydrogen, so it has a positive 1 charge. Okay. Cool. Good question. Yep, Aiden. Okay, that's a good question. Sometimes these amino acids are categorized as simply charged. Sometimes they are called polar charged. But I think what they're trying to do here is to try to keep it, they're trying to keep it more simple, but they are certainly polar because they have a charge on them, right? Okay, so anyways, good question. Other questions? All right, cool. Functions. So remember, the function of something is absolutely related to its structure. I'm just gonna check and make sure I still got. Out there, are we still good? Are we still good with sound? Give me a yell. Good. Thank you. Thank you. Thank you. Awesome. This is like the first time ever. Okay. Now this is complicated. So proteins have 4 levels of structure. 4 levels of structure. Okay. Sweet. Seems like it's all right. Okay, let's talk about the primary structure. What do we know about protein synthesis? How are the proteins made? We start out with, what's that stuff in your nucleus that tells you everything you're supposed to do? DNA. Okay, so we got all the instructions for all this stuff is in the DNA, right? Then we come in, we get what's called transcription. And the end product of transcription is what's called mRNA. Message RNA. That message RNA gets taken out into the cytoplasm. And then what happens? Bunch of proteins, a whole community of proteins. So remember, it takes communities to make life happen, right? Life doesn't happen in isolation, ever. Maybe, I don't know. Typically not. So anyways, this community of proteins gathers around, collects, boom, starts translating this mRNA. And one of the products of that is literally, it grabs amino acids and brings them together and then begins to form peptide bonds between different amino acids. Grab this one, boom. Grab the next one, boom. Grab the next one, boom. All in accordance to the sequence of the DNA, of the message RNA. Okay. So the primary structure then would be the sequence in which those amino acids are brought together. So did anyone ever, as kids, I didn't have these, my younger sister did, Lucky. No, I'm just kidding. Maybe I did, because this is like little baby toys. But if you've ever seen those little baby toys with the little plastic things and you pop them together, like the red and green and different shapes, and you pop them together and you can make these long chains. You know, no idea what I'm talking about? Okay, well, some people do. It's kind of like that. Anyways, so this is the actual sequence of amino acids. This is the order in which those amino acids are put together. Asparagine, lysine, proline, asparagine, glutamate. Oh, what's going on over here? Oh, what's going on? Legos. Oh, Legos. Legos work. That's a good one. Yes, I was being too complicated. Legos. They're not real flexible though. But yeah, you just put a bunch of Lego bricks together. You just make it, this would be like making a big long chain. Yellow, red, green, 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 yellow, red, whatever. Now they have those kits, right? They have the kits. You have to make what's in the kit. Who wants to do that? I just want a box of blocks that I can make stuff with. Whatever. Okay, so this is the sequence of amino acids. They're putting it together. It's being assembled in a straight line. Bum, 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 bum. That's the primary sequence, primary structure. Oh, every time I go to this computer, I love it. I had 2 cups of coffee today. You better look out. Whew. In addition to the 3-letter abbreviation that we talked about back here, LEU, GLY, PHE, TRP, trippy. It's trippy. Amino acids can also be represented by a single capital letter. So you may see a protein represented by just a big string of capital letters. All right? Now that kind of gives us another analogy, because when we combine amino acid monomers to write a polymer, we can also think about making words, right? For example, we can have the same letters representing amino acids in different order to form different words, like lame and meal. Now, sometimes these 2 words mean the same thing. It's supposed to be a joke. Anyways, but lame, someone who has difficulty walking. Meal, something you eat. Same letters, different order. Same letters, different order, different primary structure. Ultimately gives the word completely different meanings. Structure and function, even in words. We can have different letters form different words that sound the same. Made, I made my bed. Made. I have an opportunity to stay at a hotel, so I didn't have to make my bed. Okay? So letters form words. The words have different meanings. So this little 1, 2, 3, 4. So this little peptide, the little peptide that could, is made of 4 amino acids. It's a tetrapeptide. It's going to have a different function from one that's made of the same amino acids, but in a different order, okay? So this is one primary structure, and on the right, this is a different primary structure. So the primary structure, simply the order in which the amino acids are brought together, combined to form a chain, a peptide chain. That's the first thing that happens. So remember we were talking about way back when, was that like Monday? I don't remember. Alpha subunit of hemoglobin. Remember, this is a subunit, so this is a polypeptide. Right here. It's actually more than a polypeptide, but contains 141 amino acids. Okay, see all these letters? Okay. If these were all in one big long string, just imagine that. This is the primary structure. So as these are being put together, between the M and a V, there's going to be a peptide bond. Then the next amino acid would be bound to that. There'd be a peptide bond between V and L, between L and S, between S and P, P and A and D, et cetera, et cetera, et cetera. You got that? So the primary structure can be very long for some proteins. So let's consider for a moment a tetrapeptide, one that's made out of comprised of 4 amino acids. I discussed this briefly with someone before the end of class on Wednesday, so you're not allowed to answer this. But we have 20 different amino acids. So how would I actually determine the number of combinations that I can now get? Okay, we've got Anthony. So you take the number of amino acids, 20, and if my peptide is made of 4 amino acids, I raise it to the 4th power. Now, if we do that math, how many possible tetrapeptides can I get? This is only 4 amino acids long. 20 times 20, that's what, 400? $160,000 different combinations. Bam. That's a lot. Imagine we now start going out into one that's 141 amino acids long. Now we're going to probably get into the millions here, right? No, even much more than that. 20 to the 141st power. That'll break your calculator last time I checked. So this is why proteins can have just almost, so this isn't heresy, almost an infinite, more than our minds can possibly imagine, number of different proteins.
Sorry, Father Sean. What was
and each of them would have a slightly different structure and a slightly different function. It's why one of the reasons why our bodies are so incredibly complex. Even a cell, because all these things are happening inside of all these proteins. Most of them are actually working inside of our cells. Isn't that amazing? Hey, got it, Matthew. Hit it.
I'm sorry, what was that number one more time for the 4 amino acid combinations? I didn't get it.
So it's 20 to the 4th power.
Thank you.
And if you do the math, it's like 160,000 different tetrapeptides. And if you want to break your calculator, try one that's 141 amino acids long. Yo. Okay, questions about that? Okay, really. So the protein, as it's being formed, starts to come out of the ribosome, and the whole protein structure, this little string. And then it starts to twist and kink and form shapes, because our proteins don't function as a long string typically, right? It's more of a folded shape. So we're going to start talking about some more complex things, right? It'd be kind of like if we had, let's say we had some sheet metal, all right? Or maybe the sheet metal was made out of, well, we could say steel or aluminum or tin or brass or copper. And we made it a certain length, okay? We could sort of say, in a sense, maybe that would be our primary structure. What's your basic material you're starting out with? Now, if you wanted to make an airplane, you're going to start taking a piece of sheet metal, and you're going to start molding it into a certain kind of shape, aren't you? Maybe start curving it. Maybe some of them are angles. Maybe some of them are curves. Well, secondary structure. These amino acids. This is a bad picture here. It's a really bad picture because it's not what's actually supposed to be on the screen. And I checked this morning. And that is not. Well, isn't that lovely? Okay. How about here? All right. I can fix this. I don't know what's going on there. Okay. Okay, I was going to go back to sharing. Sharing is caring. Okay. So let's draw an amino acid. Let's use the pen. Wow, this is going to work well. Okay, so this is not going to be the most aesthetically pleasing thing you guys have ever witnessed in your life. But you know what? There's R1. What should this be here? There we go. I can't believe I'm doing this. Okay, what's this one? This is a carbon. Let's draw our oxygen here. D. Now we're going to have our peptide bond to another nitrogen. I'm really sorry about this. I did check this today. It had something different on my screen. I'm telling you it did. C, R2. Yes, I'm still going to draw my R groups on the same side. Going back to our question on Wednesday, because that's just how I do things. C, double bond O. Wow. OH. Okay, go away. All right, now here's my peptide bond, and these are all of the elements that are involved in that peptide bond. You remember, so we did talk about this on Wednesday, that these do not move. They're stable. There's no rotation around this bond. This carbon, this nitrogen, this hydrogen, and this oxygen, they remain fixed, stable, in place. But we got a lot of movement around the bonds here. So this carbon, this alpha carbon, it can move and groove, shift, can move around. Okay, so What ends up happening then? As this, so remember, the protein's being made, it's being synthesized. Amino acids are popping out of this protein complex. Part of that complex is the ribosome. They're being added one after another in a linear fashion. And this thing sort of pops out almost like pearls. We can maybe think of like a string of pearls coming out. And then it begins to twist and begins to change shape. So we're going to get to this, but some of the shapes can form. Sometimes the shapes are ribbons. Sometimes they are a helix. Boing, 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 looks like a spring. Okay? But and then I want to see if we can actually identify an amino acid. So who wants to be, I'm going to stand back. Who wants to be a brave person and come up and show us where there's a whole amino acid on this chain here? We'll go on this chain right here. So this is a continuous chain. It's actually going, what this is following, let's see here. Anyways, so who wants to come up? Anyone? Anyone? Bueller? I see a lot of heads down. Don't call on me. Don't call on me, please. Oh, no. Okay, what do I want to look for? This is a little different, right? This is different than what we've seen before because now we got a whole bunch of amino acids all linked together. I want to look for a what, and a what? Sophia. What are my two ends of my amino acids? So we got a carbon, an oxygen. Okay, that's on one end. And what's going to be on the other end? Good. Separated by an alpha carbon, right? So that's what we're looking for. Whoa, that's easier. How about there? Does that look like one? Yo, there's an amino acid right there. Can we see it? There's my amine. There's my alpha carbon. There's my hydrogen. R. And then we got my carbonyl. Okay? Where's the hydroxyl? What happened to the hydroxyl? John Cost. What's that? What happened to the hydroxyl? Quattro. What's that? It got rid of it. Good. How did it get rid of it? What kind of reaction gets rid of dehydration? Yeah, good. So it got rid of it. So there used to be a hydrogen here and a hydroxyl here. Gone. Okay, good. Here's another one. And another one's gone, and another one's gone. Another one bites the dust. Hey, we're going to form a bond. Another one bites the dust. Dun, 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 dun. Anyways, okay. Where was I? All right. I'm not going to do my Freddie Mercury. Anyways, so now we're still talking about secondary structure. How do these structures actually begin to form? So they come out in a string, and then they start to form, bend, twist, forming these shapes. Here's what happens. And we're going to do a worksheet about this next week, so this is a little bit of a preview. I'm going to back up. That's me backing up. Okay, so let's go. Let's put our polarity helmets back on. It's time to go to the polarity helmet. Oxygen. much more what than carbon? Has a lot higher electronegativity than carbon does, right? Right. What does that mean? That means that and we got two bonds here, double bond. So we're sharing, these are sharing 4 electrons between them. 2 belong to carbon, 2 belong to oxygen. But because oxygen is much more electronegative than carbon, what happens to those electrons that are supposed to belong to carbon? Kyle. They're hanging out with oxygen, which means that oxygen then has a what? Partial negative charge, right? And carbon has a partial positive charge. What about across the way? Duh. Look at this. Nitrogen, much more electronegative than hydrogen, is it? How about, can I get a yo for that? Yo, absolutely. So that means that nitrogen is going to hang on to that oxygen more, which means nitrogen has a partial. Did I say nitrogen is going to hang on to that oxygen more? Nitrogen is going to hang on to that electron more, which means that nitrogen has a partial negative charge, right? Follow the electrons. Like the smiley cookie at a pirate game, follow the smiley cookie. We know they're going to lose again because the team is really bad. What? Anyways, but that also means that hydrogen has a partial positive charge. So, here we are. Partial positive charge here, partial negative charge here. They're close to each other. What happens? What are we going to form? What are we going to form? We're going to form a bond, but what kind of bond? Ionic bond? No. Covalent bond? No. What kind of bond are we going to form? Hydrogen bond. And how would I represent that hydrogen bond? There's little horizontal dashy lines. So, oh, it actually tells you up here. Hydrogen bonding. I gave you the answer on the screen. I spoon-fed you. And you said, I do not want what you offer me. Go away, monk. No. So the answer was up here. But here's the big point. Secondary structure involves amino groups and carboxyl carbonyl groups. This is what forms the secondary structure, not the R. The R group is involved in tertiary structure, not secondary structure. The R group is involved, it's the pirate bond. It's involved in tertiary structure, not secondary structure. So, anyways, I'm fixing that. I'm fixing that. There's another example. So, material for the exam, quizzy. Stops with the primary structure. Don't worry about secondary structure. Awesome. Questions? And for next week, we are going to finish. We're definitely going to finish with proteins. I know I've been telling you that for a long time. And we will absolutely start talking about carbohydrates. So read about carbohydrates and answer, where are the questions? Answer the questions at the bottom of page 3. So finish page 3 We're going to dive into that on Monday. Awesome. Thank you. All you out there in La La Land, are you good? Was it good? You still hearing me? All right. Was that the best audio yet?
It was great. I liked it.
Okay. I think what might have been happening was I think when I tried to go over and point to the screen.
Yeah.
You have hang on a second. If I could ask some folks to help clean up again, please. Thank you. Oh, the dispensers on the walls. You guys are awesome. So yeah, I think what I'm going to do is just not. I think what I was doing was I was stretching the cord out and it was coming unplugged from the other cord. So anyways, good. I'm glad it worked out this time.
Yeah, me too. Thank you.
Oh, and I'll take care of your, you're going to, yeah, I'll take care of your stuff.
Okay.
All right, man. Take care.
You too. Bye-bye.
God bless. Okay, can I stop this thing from recording? Oh, there's how it does it.